The overall goal of this protocol is to examine ciliary mutant mouse cochlea for planar-cell-polarity phenotypes.
These techniques allow for an extensive detailing of the cochlear phenotype, which will lead to a more precise understanding of the ciliary involvement in establishing vertebrate PCP signaling. The main advantage of this protocol is that once mastered, the dissected cochlear epithelia can be used for a variety of different types of phenotypic analysis.
After preparing reagents and tissue from experimental animals, at developmental stage E16.5 to P3, use a scalpel blade or a small pair of scissors to dissect the head along the sagittal midline, beginning at the nose and extending caudally. Remove the brain from each half of the skull with a pair of forceps. Then, identify the temporal bones, which contain the developing bony labyrinths of the inner ear. While working with the sample under a dissection microscope, use a pair of forceps to prise the bony labyrinths away from the skull by running the forceps gently underneath the bony labyrinths. Following dissection, use the tip of the dissecting forceps to clear the oval and round windows and make a small hole in the apex of the cochlear spiral. Next, fix the bony labyrinths in 1.5 milliliters of 4% paraformaldehyde for 5 minutes on ice to allow for easy removal of the tectorial membrane. Now, place the bony labyrinths in a black silicone elastomer-coated dissecting dish containing PBS to facilitate dissection.